Let me go through the agenda really quickly for everyone and welcome to the webinar. So we have an exciting webinar for you today on the kidney safety biomarkers and how to actually apply those. As you all know, we've done a series of pre-recorded talks. Hopefully everyone's had an opportunity to take a look at those. If you haven't, those talks will still be posted on the CPATH website. But as far as the agenda, I'm giving a quick welcome now. We'll go ahead and do some quick introductions from our presenters to give just a synopsis around the talks that they pre-recorded. And then at the half hour, we'll go ahead and open up the session for a Q&A panel discussion. And so we have present with us Stefan, Sultana, Warren Glaab, Gary Friedman, Stephen Piccoli, and Elisa Thompson. And we'll go through basically one by one, giving introductions and then giving a little bit of background on our presentations. So let me start out. So what I did within my presentation is I talked a little bit about PSTC and PSTC's 15-year anniversary. As many of you know, PSTC is a consortium comprised of several pharmaceutical companies, academic members, and importantly, members from health authorities. Our primary goal is really around the qualification of novel translational safety biomarkers and their use in drug development. PSTC has many collaborations. We like to collaborate. That's how we get things done, not only with individuals across industry, but also other groups that are working on similar projects that we are. One of our most important collaborations and most germane to the discussion today around the kidney safety biomarkers is our collaboration with the Foundation of the National Institute of Health and their Biomarkers Consortium. What you're going to see from the pre-recorded talks and from the discussion today are advancements around these kidney safety biomarkers, and this could not have been done without the collaboration with the FNIH Biomarkers Consortium. So why do we need new advanced kidney safety biomarkers? Serum creatinine has been used for years. I think what the issue is with serum creatinine is that it has both specificity as well as sensitivity limitations. Not to say serum creatinine is a bad biomarker, it's a great biomarker for looking at kidney function. The problem is that it's not very sensitive, and it takes a significant amount of damage to the nephrons within the kidney in order for serum creatinine to actually respond. And that's a problem, because what we see in drug-induced kidney injury is a progressive loss of, or progressive damage to the kidney that would actually go undetected with serum creatinine. And then the reason is because what we have is what's called functional renal reserve within the kidney. In other words, the kidney is a very important organ for survival. So therefore, there's functional redundancy built into the kidney so that a single exposure to a nephrotoxicant wouldn't result in the death of the organism. Instead, we have numerous nephrons, and sadly, serum creatinine doesn't respond until there's significant loss. And so the solution to that has been the bringing forward a set of novel urinary biomarkers that actually look at kidney injury, just not kidney function. And those biomarkers are listed here. We put together a program in order to understand how these biomarkers perform. And this really included three primary phases: a non-clinical learning phase, in which these biomarkers were characterized in rats, dogs, and in some case monkeys, non-human primates, and in order to relate their response to histopathological change within the kidney. There has also been a clinical learning phase where we have characterized the baselines of these biomarkers and understood, the relative variability associated with these biomarkers in humans. Then there's been a confirmatory phase, which is ongoing, and that is to really demonstrate the fact that the biomarkers in the clinic do respond how we think they respond. In 2018, there was a pretty significant event, and that was the fact that our biomarkers were qualified, at least 6 of those biomarkers, as a composite measure. And that's really what the webinar today is going to focus around, especially the discussion part is figuring out how to best utilize these biomarkers and apply them within the clinical setting. And we do know these biomarkers are more responsive than serum creatinine. What we have here is a table that shows in the left column the biomarkers, in the next column the response of the biomarkers when there's medically significant increases in serum creatinine. And what we can see is that the biomarkers across the board respond when serum creatinine is increased. In red, what you can see is the biomarkers responding when there was not a medically relevant or significant increase in serum creatinine, thus demonstrating their responsiveness. On the far right column, what we see is basically the natural variability of the biomarkers in the healthy volunteers, and the biomarkers are relatively stable within the urine. Getting back to the 2 prospective studies that I've talked about before, these studies have been completed. There's an aminoglycoside study in cystic fibrosis, as well as a cisplatin study in cancer patients. These represent our prospective clinical confirmatory studies. These are currently being analyzed, and I think what we'll find is that after these are analyzed and we have discussions with regulators, we can expand how we actually apply these biomarkers, but that's a story for another day. So what I concluded in my talk was basically that, we've made significant advancement over the past several years for these clinical safety biomarkers or translational safety biomarkers. One of probably the key points that I made was around what we want to do after these biomarkers are being used by drug development sponsors. And that's what we want to do, is we really want to share that data and share the experience with these biomarkers. And what we've created is a data repository named the Biomarker Data Repository, or BMDR, to go ahead and be able to share this learning across different individuals, or across various stakeholders. So with that, I'm going to go ahead and turn the presentation over to Stefan Sultana. He's with AstraZeneca, and he's going to give a short presentation and summary on what he presented. Stefan?
Thank you, John Michael. Can you hear me?
Yes.
Thank you. Hi, everyone. I'm part of the patient safety organization within AstraZeneca, and I've been part of PSTC now across 3 different companies for the last 12, 13 years. And this is some work which PSTC has done, which resulted in a qualification of the composite measure, which is a biomarker panel. Can I have control, please, Laura?
Stefan, you should have control now.
Okay, thank you. There we go. Okay, so what is the composite measure? It's a biomarker panel of 6 urinary biomarkers, qualified recently, September 2018, for use to help detect renal tubular injury in phase 1 studies with normal healthy volunteers. John Michaels described the work that went into developing the biomarker panel in the webinar recording. It consists of 6 different biomarkers, and the important point about the biomarkers is that they reflect different states of function within the kidney, so that they represent a wide spectrum of the injury response and repair response in the kidney. The qualification itself also includes a formula for how to calculate the composite measure, and it includes thresholds that can be used in phase 1 clinical studies to help with detecting renal injury. There's an approved context of use associated with the qualification. So we really should be using the composite measure when we think that we have a risk or presence of renal tubular injury. And we also should have demonstrated positive biomarker data in toxicology studies in order to help with decision making in the phase 1 environment. We should use these biomarkers alongside standard renal safety monitoring tools like serum creatinine, BUN, urinalysis, and we should be using these biomarkers to guide cohort decisions in single ascending dose, multiple ascending dose studies. It's important to point out that the biomarker panel is not currently qualified for use to monitor individual healthy volunteers in a phase 1 studies, and also the thresholds that apply to the phase 1 studies have not yet been tested and qualified for phase 2 patient studies. The qualification document includes a decision tree to help users in applying the biomarker panel, and I'm not going to take you through this decision tree in detail. That's part of the recording that you can see online. But basically, you progress from preclinical testing of the biomarker panel. If appropriate, if the biomarkers respond in the preclinical setting, you can start using them in the clinical setting. And the biomarker tools never replace serum creatinine, BUN. They're used on top of the existing safety biomarkers. So if you find changes associated with serum creatinine, for example, you will probably make a decision to stop escalation based on creatinine on its own without having to bring in the biomarker data. The thresholds which you'll find in the qualification package have 3 dimensions to them. First of all, you choose a threshold based on your study design. So does your phase 1 clinical study have an equivalent placebo group or not? And if you think about how single ascending dose, multiple ascending dose studies are typically designed, you bring in different cohorts of individuals, and you normally have an unequal randomization of subjects to assignment of subjects to placebo and to active. Let's say, for example, each cohort has 8 subjects. It's normally 6 + 2, right? 6 on active, 2 on placebo. So in the first 1, 2, 3 cohorts of subjects, you will have a disproportionately small number of placebo subjects. You may not be able to use the placebo comparator side of the table. We'd recommend using the no comparator numbers for the thresholds. The second dimension that we used for thresholds is the sample size of each dose group. So these range from 6 subjects all the way to 20+ subjects per treatment group, and you select the right threshold based on the number of subjects in your study. The last dimension is the p-value, or the probability that you want to assign to the threshold. The higher the lower the p-value, if you like, the higher the probability that the finding is not down to just down to chance, but obviously the lower your p-value selected, it's going to take a higher threshold to cross to be certain that you're seeing a definite response, and therefore potentially you're causing a higher degree of damage to the kidney. So you probably want to focus on the 5 or 10% p-value when you're designing your initial studies. These are just some tips which we've picked up along the way of using these thresholds. You should be including some, if not all, the biomarkers in your toxicology studies to see whether they correlate with the pathology that you're seeing with renal tubular injury. If you do see this correlation, then it's very reasonable to use these biomarkers for decision making in healthy volunteer studies and phase 1 studies. We'll bring in Steve to talk a little bit later on about the assays, but I think it's quite important that when you're choosing the lab for which you're going to use to measure the biomarkers, you should look at our qualification document, because there's a table there which gives the assay performance characteristics for the 6 biomarkers which we feel are important to choose. These are the assay characteristics that were used for the qualification package, and therefore they represent the minimum performance characteristics for using these in the clinic. You're going to collect samples at baseline. Typically, you're going to collect more than one sample because you want to be able to repeat testing at a future date if you need to. And you're going to select post-dose time points based on the toxicology study, for example, or based on your pharmacokinetics of the drug, depending on how long it's going to take to wash out from the body. Once you've ran the study and collected the samples and tested them, you're going to calculate the composite measure for each of the time points for each of the dose groups, and then look across the time points to look at the pattern across successive time points. You don't want to, obviously, you want to be careful about looking at isolated numbers and making decisions based on an isolated elevation. You want to look at the pattern over time. And remember, of course, that unless there's a very good reason why serum creatinine should go up, which is due to the pharmacology of your molecule, for example, or the population that you're studying, you really should be looking at serum creatinine changes first, irrespective of any negative biomarker results. So serum creatinine basically trumps biomarkers when it comes to decision making. And I just got one slide because we also presented a couple of case studies. The first 2 studies actually that we started using these biomarkers within the AstraZeneca drug development program. And there's a couple of case studies which are posted again online for people to look at. I've just got one slide which summarizes some of the main points. Basically, we only applied the biomarker when the preclinical biomarker data correlated with the renal tubular pathology. We didn't test all the 6 biomarkers. We only tested 3 and 4 biomarkers for these 2 particular examples, and not all the biomarkers responded. So 1 or 2 biomarkers responded strongly in each of the programs, but that gave us enough confidence to use them in the clinical setting. The 2 examples include a multiple ascending dose study, which is quite unusual in that it's a 28-day dosing study. There are multiple sampling time points included in that study. We sampled at least twice per week. So in this particular study, there's about 10 or 12 measurement points per individual. And 1 single ascending dose study where we only sampled baseline and 3 or was it 4 post-dose time points. The multiple ascending dose study was the first time we're using this biomarker panel, and one of the lessons was that when you test samples, try to test all the samples from one given individual in the same analytical run to avoid a batch effect from one set of samples to the next set of samples, because otherwise you can get batch effects on the results, and you can get certain time points which stand out as potential trouble spots, which might be just due to the analytical method that you're using. And I think it's fair to say that the experience that we gained in these 2 studies gave us the confidence to keep on using this panel, and we've now subsequently used it in at least 3 or 4 other projects, and we continue to use this phase 1 biomarker panel. And with that, I'll hand the control over back to you, Laura, and invite Warren to present the Merck case.
Warren, you have control.
Thank you all. Thank you, Stefan. So yes, hi, my name is Warren Glaab. I'm at Merck & Company in West Point, Pennsylvania, and I'm the head of the Systems Toxicology Group in the Safety Assessment Department. And we've been working with the PSTC also from the very early days and working in some of the kidney biomarkers as well as some of the other target tissue toxicities. And today I'm going to present just a quick sort of case study of how we've actually used some of these kidney biomarkers, and it's really the success of leveraging those data preclinically and translating into the clinical setting. I have control. I cannot advance, Laura. Oh, there I go. Great. All right. So So at Merck, we've actually used these biomarkers, these novel safety kidney biomarkers, almost from day one, from the early days of the PSTC. And we've really deployed them in numerous development programs here at Merck. And it's really the use of these biomarkers to both demonstrate the monitorability of the drug-induced toxicities, as well as the reversibility of such lesions. And using these biomarkers has really helped advance several different programs, not only in the preclinical space, but into the clinical settings. Right. So the example I'm going to give is one of the early ones, and this is relebactam. And this is an example of maybe 10 years ago when we started using these biomarkers to monitor the toxicity and to try to help leverage those data sets to move into humans with our compounds. So here you just a picture of the nephron, right? So here are the biomarkers that inform different toxicities within different segments of the nephron. And we have many different programs at this point that have affected different segments of the nephron, most of them predominantly in the proximal tubules. And the example that I'm going to give today is this, it's a BLI compound, right? And it's relebactam. So relebactam, it's a beta-lactamase inhibitor. And it was originally developed as a fixed-dose combination with Primaxin. And it's really for some of these pretty severe infections, right? So, bacterial pneumonias as far as some of the intra-abdominal infections and some complicated urinary tract infections. And so it's noted, right, so this is an old example, right? And this predates the composite measure that Stefan was just talking about. And it was at the time when we were looking at several different of the biomarkers to see if we could actually monitor the induced toxicities in our studies. And this particular example demonstrated toxicities in kidney tubule in non-human primates. Okay, and what we did is we actually looked at the several different biomarkers to see if we could correlate with the histopathology. And KIM1 actually demonstrated superior monitorability of that induced lesion in the non-human primates. And then we also used that biomarker in some of the preclinical studies to demonstrate the onset of the toxicities as well as the reversibility of the lesion. It was then that we actually used these KIM1 data, far more sensitive to BUN and creatinine in this drug-induced tubule injury, to move into the clinical setting, right? And we ran KIM1 in our early phase trials, the phase 1 trials, and what we found was a lack of response of KIM1, right? So no increases in KIM1. Built confidence that this lesion was maybe not relevant to humans and were at a sufficient margin to really advance the compound in the clinic. And the compound then has proceeded through phase 2 and phase 3 trials. We did not run KIM1 in those later phase trials, but just continued to monitor BUN and creatinine. And really what we found was that there really was no signal at all whatsoever for kidney liabilities with this combination compound. So, ultimately, we received approval for the drug in July 2019. So it's just a really good example of how even some of the work that we did 10 years ago really has impact in getting the compound through not only preclinical and clinical development, but ultimately to approval in the clinic. Gary, at this point, I will turn this over to Gary Friedman.
Thank you very much, Warren. Warren, can you confirm you can hear me?
I can hear you. Yes, I can.
Excellent. Thanks so much. And thank you to Critical Path for inviting me to be a participant in this panel. It's really been an honor and a delight to be a Pfizer representative on this project over the past, I guess for me, it's been about the past 8 years. And as I'm going to build upon what John Michael, Stefan, and Warren have already presented, as John Michael indicated earlier, The qualification determination letter was issued in 2018, and really our goal now is to continue to advance the validation of these biomarkers and the applicability of these biomarkers. And the foresight that has been demonstrated at the Critical Path Institute in terms of how to really begin to pool all data going forward really rests in a mechanism here that's called the Biomarker Data Repository. So the BMDR is essentially an anonymized, safe environment where data from studies utilizing the biomarkers that Warren and John Michael and Stefan have been describing, utilizing those data from various companies, from academic centers, and other organizations, pooling those and placing those into the Biomarker Data Repository has the potential to offer us novel directions. And that's what I want to just highlight now, and then just end my part of the talk on where these might have some exciting applications. As John Michael originally alluded to, we have now completed 2 confirmatory studies utilizing the full panel of biomarkers. And these data are currently being analyzed and adjudicated and will be submitted to FDA for further qualification. And what we want to highlight here is, number one, we don't have the output from these 2 studies yet. All the data are still blinded, but we know what we will have at the end of it is about 120 patients treated with a known nephrotoxin, either an aminoglycoside or cisplatin. Somebody is just checking to see, can I still be heard?
Yeah, Gary, I can hear you.
Okay, great. Aliza was indicating there was some issue with sound. So going back to the point I was just focusing on, so at the end of these first 2 studies being completed and the data ultimately ending up in the Biomarker Data Repository, we'll have about 120 patients that have been treated with a known nephrotoxin. We'll have about 60 patients treated with a regimen that is not known to be nephrotoxic. We're aware, however, because of the opportunity to participate in other biomarker consortia, we're aware of data within that were generated within the European Union. So going back to the mid-2000 teens, we know that there were 3 different data sets that were generated with patients with malignancy treated with cisplatin, with patients with a known cardiovascular disease that were then exposed to IV contrast medium. And we also have a non-disease state. So we had 39 normal healthy volunteers. So we know that we can advance if there is a way to comply with all the EU data privacy legislation. If there is a way then to have these anonymized data then pooled within that same biomarker data repository, we have the ability now potentially to go from 2 disease states, so malignancy and cystic fibrosis with pulmonary infection. We now have the ability to advance and now include a 3rd disease state, patients with cardiovascular disease, and now advance to a 3rd potentially nephrotoxic drug, and now advance from the 120 cases now to up to 530 cases, advance from the 60 controls up to nearly 120 controls, and now introduce the normal healthy volunteers. One of the nice things about these 2 consortia is that the IMI Safety and the PSTC consortia, the biomarker panels that they independently converged upon, have great areas of overlap. So the majority of the PSTC biomarkers for renal tubular injury are also contained within the IMI Safety renal tubular injury biomarker panel. As you just heard from Stefan, as well as from Warren, each of us that have been participating in this project as representatives of our individual companies, we are aware that that drugs in development can now be moved forward since the qualification of the full biomarker panel. We're aware of other drug companies that would like to utilize these panel of biomarkers, the full panel. And currently, these are anonymized. There are two different pharmaceutical companies that are conducting first-in-human studies using the full panel of biomarkers. And they will be assessing internally their individual levels of change within the biomarkers, along with the standard safety biomarkers of creatinine and BUN and serum cystatin C. So all of these data, when these become available, will take us now in the biomarker data repository, could take us to having 5 disease states, 5 potentially nephrotoxic drugs, and now advancing to beyond where we were before. So quite a jump in terms of the number of patients treated with a nephrotoxic agent and then the full panel of biomarkers being assessed against controls and having the normal healthy volunteer data. So becoming a very robust environment. In parallel with all of this, we know that the assays that have been developed for the panel of biomarkers, we know that those assays have become available more broadly. And what we're in process of doing now is a literature-based search of publications wherein the biomarkers have been assessed, whether all 6 or some subset of them, utilizing the panel of biomarker, using the same vendor or different vendors providing those assays. Those are now being assessed across a broad swath of literature to see if there are data that could be pooled into the biomarker data repository as well. And finally, an additional opportunity for growth of this biomarker data repository lies in 2 studies that are currently being conducted within the EU. So there are 2 through TransBioLine. There are 2 studies that are ongoing looking at glomerular injury biomarkers. And in conjunction with those glomerular bioinjury markers being studied, the renal tubular injury biomarkers that we've been focused on today are also being assessed in these patients who either have a malignancy being treated with anti-VEGF antibody or in patients with a high risk of developing preeclampsia. So ultimately, beginning with the 2 confirmatory studies wherein we'll have about 120 cases, about 60 controls, you can see that we already can be really jumping these numbers up to really look at, is there consistency in terms of the thresholds that have been set early on? Do those change over time? In terms of the composite measure, does that change over time as the size of the database grows? So a lot of opportunities for growth here, and we're very, very excited about this. Essentially, a leave behind that biomarker data repository becomes a universal translator for looking at the effectiveness and the detection capability of either individual biomarkers or the entire panel or subsets of those biomarkers. Ultimately, where we want to go, though, is to take this and move from where we are now in terms of the use of the full panel for assessing cohorts of patients, of healthy volunteers being exposed to potential nephrotoxins. we'd like to also begin to understand that just as healthy volunteers have baseline levels of these biomarkers detectable in their urine, we would also like to understand if you take populations with chronic kidney disease at various CKD levels, if you take diabetics or pediatric patients or geriatric patients or oncology patients, if we can begin to look at what are the baseline values for patients with in any one of these special populations, can we then look at the change of the biomarkers over time in response to exposure to a nephrotoxin? I apologize if I went overboard here a little bit in terms of time, but really appreciate the opportunity to present to you how we see the potential future development of this biomarker data. And I think we'll now be moving over to Dr. Elisa Thompson from FDA. Thank you very much.
Hey, thanks, Gary. And also thanks, Stefan and Warren, for your presentations and summaries today. I think that was really helpful for setting the tone. What we'd like to do now is really move into the question and answer and panel discussion session. And as Laura reminded you, if you do have questions, you can raise your hand and verbalize a question, or you can go ahead and put it in the Q&A section of the chat to the right. But what I'd like to do first is introduce Elisa Thompson. And Aliza, if you can give a quick introduction of yourself and then kind of give us your thoughts around the composite measure and this project overall. I think that would be really helpful.
Sure, happy to do so. My name is Elisa Thompson. Hopefully, John Michael, can you hear me?
Can hear you perfectly, Aliza.
Great. And I work at the FDA. I'm the Deputy Director of the Division of Cardiology and Nephrology in the Center for Drug Evaluation and Research. And I have had the pleasure of essentially since joining the FDA of working with, I think, most everyone or many of the people who presented today and learning from them, and really shared the excitement in terms of seeing these biomarkers advance. I want to thank, CPATH, John Michael, you all for putting this talk together, because I think it's really, or session together, because I think it is really critical for the community to hear how far we have come. I also just want to thank as well the speakers, because I think they've highlighted some, or provided some wonderful examples of how they've used these biomarkers to inform decision-making in their own companies to de-risk products, or to figure out if they need to, go back and do further work on a compound. But actually where I want to focus my comments, John Michael, not just acknowledging the important work that's been done and the importance of the qualification, but just to maybe make a comment or two about use beyond the qualified setting, which is really the idea here is a translational setting in phase 1 studies using this composite measure, so all of these biomarkers. And the reason why I want to make a few comments outside the setting is because the reality is the qualification is an important step forward and provides an important pathway to use these biomarkers within a certain setting. But the reality is, these biomarkers, subsets of them are being used outside of that setting in drug development to inform decision making. And I just wanted to highlight that. I want to highlight that the reason this is because there's a body of evidence that supports them, supports using them outside of these settings to inform decision making. It's not to say that they're perfect or that we understand everything about them, but I think as everyone knows, creatinine provides a lot of value, but it is in no way perfect. And yet we rely on that to make decisions in part. And so they're already being used within the context of the other data to inform decision making. We're seeing them used both in phase 1 studies as sort of individual biomarkers or a subset of these biomarkers. We're seeing them used in settings where they didn't necessarily study them in the preclinical setting. We're seeing them used in phase 2 studies. Sometimes, you have a drug with a hemodynamic effect, and the sponsor wants to get a better sense of, I think, whether they're the rise in creatinine, that's all they're seeing, or whether they're also getting a signal with any of these markers of toxicity as they explore the different doses. And so I think a big issue for us moving forward, we very much, heard from Gary about the future and the promise it holds. I think as we look to the future and the promise it holds, beyond the sources of data that Gary outlined, I think the reality is we already have a wealth of data. We're just not using it, and that's the data that are being accrued and the experience that's being accrued in these development programs. That's essentially not being shared, and just want to point to the biomarker data repository as a wonderful place for those data to go and to really advance our understanding and really facilitate our ability to expedite the development of treatments that Americans need, as well as ensure the safety of participants in trials. And so with that, I will stop. Again, just incredibly excited to be here today, and want to thank you all for the work you've done thus far.
Thank you, Aliza. Really appreciate your comments. What I'd like to do is also now pull Steve Piccoli into the conversation. Steve, can you introduce yourself? And then also, Stefan brought up a really interesting point, and I know that you've been, shepherding, our advancement around how we analyze for these biomarkers and actually quantitate that. And if you can just, introduce yourself and give us a bit of your opinion around that, I think that would be really helpful. And I think that would kick off the panel session nicely.
Certainly. Thank you, John Michael. My name is Steve Piccoli. I'm currently at the Sun Pharma Advanced Research Center in Princeton, New Jersey. I have had the pleasure of working with the PSTC for a shade over 14 and a half years on this project now, since its inception, selecting biomarkers, the initial assays, the normal human studies, on through generation of the full clinical data set. So it's been a long, strange trip, as I've heard someone else say. But a few things that I would like to put out here, I've been in charge of the analytical part of this, the assay validation, the data generation, and analysis. This is something that evolved very greatly over the period of time, because the initial studies were carried out nearly a decade ago. And one of the things to remember here, and it's a critical piece of information, is that while all of the biomarkers that we're talking about today have been qualified by the FDA, this says nothing about the analysis that derived the data. They are not 510K cleared nor PMA approved assays. Everything was done RUO fashion to generate the data with validations that we thought were appropriate at the time. So the analytics for this have evolved very seriously over the time of these studies. And we were fortunate that we had no major problems with reagents, with equipment that you might think would have happened over a study of this duration. But we did learn a lot of valuable lessons. We started out with what are normally considered PK parameters for biomarkers, which was the state of the art back in the last decade. And this has evolved greatly. A lot of what we've learned has been put into what is generally called the critical path white paper on evidentiary considerations for biomarker qualification. And this has allowed us to make much better judgments about how we can actually use these biomarkers. What are the important analytical parameters that we need to use to generate this data, and how that can all play into a drug development process. So I think that if there are specific questions, I might address those from an analytical standpoint a little more in detail. So I'll stop here and see if any of those pop up during the discussion.
Thanks, Steve. Thanks for the introduction and pointing out those various facets around around the analytical. We do have our first question from the audience, and that's from Tanya Zapka from Genentech, and she's I think this would be a great question for Stefan. I don't know if you can see it in the chat there or not. Likewise, Gary and Warren. I mean, this is a question more around asking the applicability of the biomarkers and your experience and how the composite measure responds to various disease states versus healthy volunteers in the absence of toxicity. And number two, experience on how the composite measure responds when a drug is metabolized primarily in the renal tubule versus tubular toxicity.
So short answer, Tanya, is no and no, but I'll expand a little bit on that. So I think we know enough about the biology of the 6 individual biomarkers to be able to predict that what we see in the healthy volunteers may not necessarily translate through one-for-one for the patient situation. So the thresholds that we use in volunteers may not be applicable for patient studies. That's why we're studying them separately. Composite measure and drugs which are metabolized in the kidney, I suppose that would probably be relevant in a few situations, right? So we know that one of the biomarkers, at least, cystatin C, and also albumin, when the patient qualification comes through, these 2 biomarkers are functional biomarkers. So you could envisage some situations where your drug is somehow interfering with the uptake of these proteins into the renal tubular epithelium to be then metabolized and cleared from the urine. So you could interfere with the clearance of these drugs, and they could trickle out into the urine in increased concentrations as part of the pharmacology of your drug. And you could probably also look at certain situations where your drug may accumulate in the kidney and not necessarily cause toxicity, but it could somehow cause an increase in NAG expression, for example, increase in lysosomal expression along the brush border, which then spills out into the lumen. So it's not a straightforward injury response of NAG being released by the cell bursting, but it could be an overexpression because of a reactive response to, for example, antisense oligonucleotides. We know that these accumulate on the basolateral side of the epithelial cells, and they do somehow, to some extent, disrupt the cellular mechanisms of the tubular epithelium. So they don't cause toxicity per se in most cases, but they can interfere with some of the cellular processes. And I think these have to be considered on a case-by-case basis.
And John Michael, just want to add something on top of what Stefan has already said. So in the first part of Tanya's question, we are aware of data sets in the setting of diabetic nephropathy, as well as in renal transplantation, where the biomarkers have been analyzed. In order to have access to that information and put it into the biomarker data repository, since those data are coming from outside of the US and coming from the EU, all the necessary agreements in terms of data protection would have to be navigated appropriately. So the answer is that those data do exist. And as Aliza was alluding to, we really do need to start utilizing this biomarker data repository as a way to really pool knowledge, to really accelerate the learnings here.
Thanks, Gary and Stefan. Any other points that the panel would like to make before we move to another question? Okay, great. So we do have another question in the chat, and that's from William Regan. The question is around, do you need to assess the whole panel as time goes on? And I think, Warren, you brought up a really good point with your lactamase inhibitor, where basically you concentrated on a single biomarker. So would you like to talk about the thinking there? And then Aliza, I'd love to get your opinion around that too, around as we learn more, how do we adapt and monitor? Oh, you're on mute, Warren, I believe.
Yeah, thank you, John Michael. Yeah, first and foremost, I think that, as I noted, the work that we did really predated the composite measure qualification, right? That said, though, we did look at a panel of biomarkers in the preclinical species, and really what we found was that KIM1 was superior, right? It actually outperformed the other markers, and it really was kind of locking into that one biomarker that just seemed to really work well for this particular induced lesion in non-human primates. That it was a decision to use only that in both preclinical studies and then the early clinical trials. You know, I would think that that's also a case by case, that for development programs where you're looking at a panel approach preclinically, if there are multiple biomarkers that respond in unit and together, right, that you would consider using a composite measure. But if one or two really truly outperform, I could see how that would then maybe bias your decision to look at just those individuals moving into phase 1.
Thanks, Warren. Aliza, any thoughts?
Yeah, and I think, you know, some of the focus around the composite has been, you know, I think both to learn about these biomarkers. And I think what you're hearing here as well is not just that PSTC and as a community we're learning, but some of the people who are on this call are clearly from pharmaceutical companies that are interested in developing internal learning, right? And so really a lot of merit to looking at all these. A lot of what we see in drug development, especially when we're dealing with compounds that perhaps have more toxicity, and sometimes are being developed for conditions or questions of toxicity, and are being developed for conditions where maybe there's not a whole lot of tolerance for risk. You know, they have cost considerations, and they're not looking to develop a large internal understanding of how these perform for a bunch of compounds, right? They have a compound that has a certain profile, and they want to advance it in an efficient way and make decisions in an efficient way. And so we are already seeing these used as just sort of a selection of these in very different ways than what the composite qualification proposes, which is appropriate, right? We have, again, a body of science around these, and we need to use that to the best of our ability to make decisions now to address issues we're seeing now in drug development.
Thanks, Aliza. A question for Stefan. So this is asking around, and how many studies have you successfully used these biomarkers, number one, and then also, did you ultimately see changes in serum creatinine, or did you didn't see renal toxicity at all?
So AstraZeneca's now has been using it for probably a couple of years now. Where are we? 2021, 2018? Yeah, probably a couple of years. We've used it. COVID took its toll on phase 1 studies, at least in our portfolio. So we took a hit because of COVID. Things slowed down quite a lot last year. But I think we've used it in about 5 or 6 different programs now, 1 study per program. Most of them are single ascending dose studies. One of them was a multiple ascending dose study, which I presented as the first case example in the recording, which people can go back to. There was 1 or 2 studies where we saw twitches in the biomarker panel as we escalated the dose. So we started getting worried that this is starting to look at it a little bit like a signal. In those 2 studies, we saw no changes in serum creatinine, no urinalysis changes, no hematuria, proteinuria coming through, nothing that would trigger a dose escalation stopping criterion, basically. And in both those cases, we were sufficiently worried that we went back to the lab. Both of them were not the parent drug. They were probably related to the formulation excipients that we were trying to use to improve the bioavailability of these compounds. So luckily the compounds are still in play, but the formulation that we're working with for those 2 particular compounds, we've had to go back to the labs for. So the short answer was half a dozen studies, give or take, and no serum creatinine changes for any of the programs.
Great, thanks, Stefan.
John Michael, can I come in
please.
on the previous comment about going about not using the full panel set and reducing the number of biomarkers perhaps? And I think this is something which will evolve over time, because we've done a certain amount of work which resulted in the qualification based on a certain data set. And the composite measure is based on within subject variability in healthy volunteers for these 6 biomarkers. So the thresholds which we have in the qualification package apply to a specific set of circumstances. If we start using less biomarkers in the panel, then you're going to start having blank data in the composite measure. You have to substitute, fold change from baseline equals 1 for all the time points for the absent biomarker, which means that the thresholds are performing differently to their intended use. But I think as companies go on and generate more data, and as we start pooling data, especially placebo data, in our biomarker repository, we could go back and reanalyze data and start coming up with some new stats rules to say, if you look at this set of 4 biomarkers out of the 6, perhaps this is the formula you should be using, this is the threshold you should be using instead.
Thanks, Stefan. And what I really like is, that this panel clearly is well educated behind these biomarkers, simply because, we're looking at what that next step is. I mean, we're trying to figure out how to best apply these biomarkers in different drug development situations. And I think that's the important part, right? There's no single recipe on how these biomarkers can be applied. Instead, it's probably going to be very programmatic, right? That's where I think we're arriving. And so therefore, the conversations with the health authorities are going to be really important about what conclusions are being made and why. And so it's just not, because these biomarkers are novel, I think there is going to be some of this experimentation with them and how they're being applied. So really good points. Any other points that folks want to make around that before we leave these subjects? So we're getting a couple questions in, but one thing I do want to do is I want to pull Steve Piccoli in for a second. And Steve, I mean, with all these different assays that are available out there, and based on the comment that Stefan made about making sure that you don't have you avoid run-to-run variability or batch variability. I mean, any recommendations to the audience how to best, utilize these assays that are currently out there?
So the run-to-run variation, is a bit of a problem over years of doing these studies. But what we found is that a properly validated assay with everything taken into consideration should be able to perform well enough to give appropriate performance for the composite measure. We have detailed some of this in the white paper. But suffice to say that it's also important in doing repeated measurements to take into account the intra-individual variation, which is another source of variation that can happen here. And if we're doing a composite measure, which is a fold change from baseline, that has to actually be a part of this. So the analytical specs that we set up for this must take into consideration those values because they play into the overall reproducibility of the results of the composite measure or the individual measurements for a single biomarker. It's important, very important to understand that in a greater sense than doing the analytics 2 or 3 times as is frequently done and being able to establish confidence intervals around the values that you generate. A reference change value for subsequent analyses is an important thing to determine whether or not that change from the prior patient measurement is actually legitimate and significant. So this requires a little bit more of putting on our scientist hat and fleshing out what really are the weak points and strong points of the assays before we go about measuring the patient samples, because the errors that accumulate there can and will affect the outcome of something like the composite measure.
Great, thanks, Steve. I think a couple things that I know were talked about within the pre-recorded presentations were ways that we tried to avoid variability, and we tried to allow different assays to be used. You know, for example, and again, I'm going to state a couple obvious things here that the panel knows, but maybe that could drive a little conversation, is first of all, we're looking at change from baseline for a given patient before and after drug. Right, and so therefore that individual, or placebo, so therefore that individual acts as their own control, and so that's a way to decrease variability. We're not looking at the absolute values and being compared. We also go ahead and take into account volume changes by basically correcting for urine creatinine, and so these were a couple pieces that were added in to decrease variability. I mean, are there other things that we're learning about as we utilize these biomarkers that we haven't taken into account or really talked about broadly, either from your experience, Stefan, Gary, or Warren?
So if they're not going to, I will actually interject here with a quick one, is that none of these biomarkers were measured fresh. And that means that stability of the samples is a very important criterion for the final outcome. And that cannot be overlooked, as everyone on the panel knows. So there are stabilities for these biomarkers. They will never be measured fresh. And the composite measure was put into place with biomarkers that have been frozen once for analysis as soon as possible after collection, but with a distinct time period.
That's a really good point, Stephen, just to go ahead and clarify that. So, you know, if you think about the conduct of a phase 1 study, these samples are not going to be measured at the site. Instead, they're going to have to be frozen and shipped to an analytical site. And so therefore, all the qualification work that we did was on once frozen urine. And so that does throw a bit of an analytical wrinkle in there that everyone needs to consider as they're moving forward. Any other comments here around the analytical or sample handling?
Just one from the internal Pfizer perspective, speaking to Steve's point, part of, although we didn't present any of the cases where we've analyzed the full panel in our early clinical development programs, the fact is we have, because we want to see exactly this, what happens when you analyze right away versus when you analyze, say, 4 or 6 months later. So lots to learn, and we have continued to commit to evaluating the entire panel so that we can generate the composite measure. We're not ready to start cutting corners yet internally from our perspective.
Great, thank you.
The only
John
thing is
Michael?
that,
Yeah,
sorry,
please.
Gary, sorry.
Yeah, and John Michael, I see a question from Mina Parikh to Aliza. So after Aliza gets a chance to respond to that, I had something I wanted to add on to that as well.
So Aliza, let's frame this question. A couple years ago, during a discussion on biomarker evidentiary expectations for use in drug development and qualification, you know, we talked about the kidney safety biomarkers, and one of the points that you brought up is that you would like to actually recommend the use of these biomarkers, right? To go and that you would like to go ahead and provide drug development sponsors with this information. But I think what we've seen is that companies haven't overtly been sharing within the BMDR. And so any other ideas around how we better incentivize or get companies to share that important data with us?
Good, and just to maybe clarify or expand, I think, early on with the qualification, so the idea of qualification is never a static process, right? It's a snapshot at one point in time of where the data are and how robust they are, right? We're qualifying it in a setting where it's a narrow context of use, where really the data are strongest. And ironically, even in the process of qualifying it, the science is still advancing, right? And so, in what you realize, as you look at the world externally and also what you're seeing internally is that, again, these companies are using these biomarkers, in terms of how many are using it. It's hard to say. We've tried to look at this issue. I can't tell you. We don't, to be honest. I think still have a good knowledge management system in place to track this, not just how many are being used, but how they're being used to inform decision making. But I know that they're being used. I see them being used. We see them being used. And so I think this sort of led to this thought, we will just, we will have this biomarker data repository, and this is the solution here, a place where all these data could go, right? I think I once heard the phrase, or I've heard the phrase, if you build it, they will come. And I think I naively thought that, all it meant was that, or all we had to do is build this biomarker data repository, or CPATH would build it and they would come. But what we quickly figured out, and I think CPATH figured out, is despite building it, despite actually providing, I think, a lot of support and trying to understand, all the support that industry would need to contribute data, they still didn't come like we thought they would. And so the next thought here was, well, what more can we do? And so, obviously, Janet Woodcock has been very interested in biomarker development and recognized their importance as tools in drug development. So she had put together a letter, I think, essentially encouraging, data sharing, and also so the thought was internally that we could perhaps, obviously we can't tell CPATH what companies are collecting data on biomarkers, but what we could do is reach out to some of these companies and include information about the data repository and this letter from Janet. And we did do this, at least for some of the products that we were aware of that fell within our division. This is hard, right? Because it means you've got to, it takes a lot of internal resources to do this. You got to convince the project managers who already have a ton of work to reach out to someone. And so we did this, didn't do it across the agency, didn't certainly do it across, the Office of New Drugs, but didn't really, at least where we did it, not clear that that it resulted in much, which is, again, this whole go back to the drawing board. So where are we now in terms of going back to the drawing board? I think what we realized is that we really needed to engage the larger community. And so we really have two initiatives in place. One initiative is led by the Kidney Health Initiative, which is a memorandum of understanding between the FDA and the American Society of Nephrology. And they're taking on a project to develop a roadmap for the development of biomarkers of acute kidney injury. And this isn't just limited to drug development. Obviously, there's or safety biomarkers. Obviously, this has implications from a drug development standpoint in terms of developing therapies to treat AKI, acute kidney injury, as well as use in the clinical setting. And then there's another really wonderful initiative that we're working with CPATH on and NIH and the patient community on, which, John Michael, maybe you could tell everyone more about. But we're hoping that really between really where we are now, the data that we're going to be getting shortly that Gary outlined, the data that are being accrued in these development programs, and frankly also the Kidney Precision Medicine Project, an initiative by the NIH where they're actually biopsying patients with acute kidney injury and doing deep phenotyping. There's really the sense that we're at a tipping point, and that if we come together as a community, that we could really, vastly accelerate and really bring to fruition what I'm going to say is, I think, the dream of everyone who's been involved in this for a long time, which is to actually have some biomarkers that are, well established as biomarkers of tubular toxicity, and that provide much more insight into where the toxicity is, how to monitor for it, the severity, et cetera. So,
Thanks, Aliza. Gary, I think you wanted to say something.
I did. Elisa, thanks for setting the stage on that. And just so you know, although you built it and maybe they didn't come right away, they are on the way. And the collaboration, like what you described with American Society of Nephrology, this is all gathering momentum. And I have, as one of my responsibilities within our organization, I'm the clinical co-chair of our Kidney Safety Council, which looks at kidney safety across all development programs within Pfizer. We are making changes in the environment internally so that this type of data, as it's accruing and as we educate teams internally on the potential value of these, more and more teams are utilizing the biomarker panels. And we are now driving senior leadership level. We are driving the desire to utilize the entire panel and then share it through the BMDR. So never give up, never surrender. It is going to come.
Thanks, Gary. And to follow up on what Aliza said about the acute kidney injury working group that CPATH is leading along with the FDA and other stakeholders, I think Aliza actually explained it nicely. It's really an opportunity for us to take all of the different activities that are going on within this space and try to pull it together and create more awareness. You know, talking with, you know, folks from NIDDK, folks in academia, there's really not awareness, and even in industry, there's really not a lot of awareness around what we're doing with these translational safety biomarkers. You know, I think folks within PSTC, I think we take it a little bit for granted that we have this great data set that links the biomarkers response to histopathology, and then it translates that to the clinical situation. Nobody else has that data set, right? Nobody else has that data. So how can we best share that data, right? And then attract other folks to share their data. Aliza brought up the KHI project around the roadmap. Luckily, I'm involved in that. Likewise, we have the Kidney Precision Medicine project, which many of the AKI working group members are also a part of. And so how do we pull this all together to really take the next step? And then I think one thing we also need to think about, it's just not drug safety, right? These biomarkers can also be used as diagnostic or pharmacodynamic or prognostic biomarkers for acute kidney injury caused as secondary to another disease, right? The disease itself, not just drug-induced AKI. And that, sure, that's going to take a lot more work, but I think that's where we can go. And that's also really important data to understand around the way these biomarkers respond in these other cases, non-drug-induced AKI cases. So I think there's lots of opportunity here, and that's the way we need to think, right? How we better talk about what we're doing here, the importance of it, and how we pull stakeholders together to achieve these end goals. Any other comments from folks?
I think I'd just like to echo what Gary said, which is that we've had these biomarker, the biomarker panel has been qualified for what, 2.5 years now. COVID took a year out of that timeline, because certainly in our company we were performing less than half the number of phase 1 studies we would normally do. And remember, this particular biomarker panel is qualified for healthy volunteer studies. For companies like AstraZeneca, where more than half the portfolio is actually oncology studies, that's not relevant for this particular biomarker panel. So, but the studies are coming, and it might be a career limiting move on my side, but I will make sure that the 5-6 studies which we've conducted, we're able to share in an anonymized way into the repository.
Thanks, Stefan. So there is one more question that's in the chat, and it really deals with what we've talked about previously, but I think we should explore it a little bit, and that's from Frank Dieterle. And where Frank's going is that, look, you guys are putting all these rules around the biomarkers, is that really the way to go? I think we have to be transparent here, right? I think one of the reasons that we did have this early qualification for the composite measure was to really get this information out around the biomarkers and put some relatively strict structure around how these biomarkers would be used so that we could begin to gain understanding around these biomarkers, right? And take that data, possibly contribute it to BMDR and really learn, right? So that when we got to the point, that evolution point that Aliza brought up, that we could then figure out exactly how to apply the biomarkers. And so I think what we're doing is we're learning here, and that was really the reason to put the structure around the biomarkers, not that that would be there forever. The way we use other safety biomarkers, we do use them in different ways, right? It's not a very highly refined or defined pathway that they need to be applied within. So I'd like to know if the panel has any additional thoughts around that, and did I say that right?
So I'll take a first stab at it, and it's great to hear Frank is on listening. I think implicit in Frank's question is the fact that he and many others have been looking at subsets of these biomarkers on their own for quite some time and have great insight into their performance. And I think that it again bespeaks to the need for the biomarker data repository to be utilized. There's so much experience out there that is not being pooled, and everything that we can do to get people to participate in this, and their data are protected for certain. But the more they share about the biomarkers that they already have great confidence in because of internal work, the more they share that, the more is the likelihood that when we do look at subsets of the full panel, that we might be able to see the ability to select down. So I'm glad Frank posed the question, and I'll stop right there.
I'd like to add in on that, if I might, because it's not only important to have the data in the biomarker data repository, but Frank's question goes directly to comparability of data generated at different laboratories by different technologies with different kits. And as we all know, antibodies may vary. So for a number of these, which were RUO kits, we're not certain that we're going to be measuring the same epitopes from one lab to another. So I think it is of extreme importance to have not only the data in the biomarker data repository, but the analytics behind how that was measured, because that will vary from site to site, and that will have an impact on the final data set and composite measure. And if we are going to answer this question appropriately, we need to understand how those things will vary and what sort of considerations we must take into account to make a final composite measure valid. And remember, this is all dependent on exactly one lab doing one set of assays with no major critical reagent changes over the entire spectrum. There is no interlaboratory variation. All of those things will come into play as soon as multiple data sources are integrated into the database.
The one thing I would add, Stefan here, so the one thing I would add to this discussion is that we've tested different labs, and one lab, which was using a multiplexing approach, we found that a lot of our samples for, I think, 3 out of the 6 analytes, a lot of them came back as below limit of quantitation. So therefore, these subjects add nothing to the value of the formula, because your baseline is BLQ, and most of the post-dose measurements are BLQ as well. How do you use that in the formula? So you have to make sure that the lab you're measuring, at least you're measuring at the same lab across all time points and change from baseline, right? So it's important that the measurement range that the lab is reporting for those biomarkers and the precision of measurement for those particular biomarkers, those are probably two important attributes that you need to watch out for, especially when selecting your lab. But I think you need to be careful that the lab can actually measure samples which are in the measurement range for your particular population.
Yeah, and if I may, I just also want to make a comment about this issue because I think it is a critical issue and it's something I worry about, especially sometimes, you know, obviously, you know, the people on this panel have tremendous experience and insight into these issues. But again, a lot of times we're dealing with pharma that, again, they have one or two compounds and they're really focused on it. And this is not the main question they're trying to answer, right? If anything, it's an issue that they have to address, and we're recommending these biomarkers to them, and they don't know anything about them. And so, at least when the recommendation is coming, I think, and we're the source of it, I think often we'll refer companies to the PSTC. With the understanding or thought that they can talk to the PSTC in some, anonymized way, or, just get additional information on sort of best practices. There's obviously also information that the PSTC has posted related to this, but just to get further insight. So that's one point I want to make. But it also circles back to this larger issue of leveraging the data from these clinical studies. I think that it is critical that, or a critical outstanding question is how good these data will actually be because of, how much attention was paid to the assays and, issues with the sample collection, et cetera. And so, obviously it was critical that the BMDR be set up so that, it elicits and you're collecting that information as well. But I think that is also just an outstanding question in terms of what's going to be out there to leverage.
Thanks, Aliza. Warren, any comments around this?
Oh, no, I just think it was well covered by the panelists. I just think that everything everyone's saying is just critically important here. Building up the data sets, it's going to allow us to make some, conclusion about performance of the individual biomarkers, the composite measure, the analytical aspect. All of these things are just really, really important. And it's over time, I think we'll just learn more, not only just about the individual biomarker performance, but if there's modifications of that composite measure and or, single measures that actually work as well, right? So it's just building data over time and having a kind of a comprehensive analyses downstream.
Thanks, Warren. There's 3 points that I want to make. Number one, within the BMDR, we do ask for the analytical information and the performance characteristics of the assay, because that's an essential question we want to ask and understand. Number two, there is an ongoing project with US Pharmacopoeia to basically do a comparison with the samples that we've collected during these prospective studies across all of the assays that are basically available, at least as kits and some of the vendors. And so we want to do that comparison to understand what type of range, and we have done that to a point within the program itself. And I think the third point is, and Stefan touched on this in his talk, and it's really important. We have a user's guide. We put that together for these biomarkers, right? And how to use the composite endpoint. Utilize that user's guide, and if you have any questions, here goes your experts. Right? Ask these guys how they did it, right? And funnel those questions through CPATH. We can get Stefan involved, we can get Gary involved, we can get Aliza involved, etc. and Warren. So any final comments from the panel, because I think it's time to wrap it up.
Glad you raised that, John Michael, because that's kind of like going to be the Rosetta Stone, when we do run those samples across the multiple different assays. So people will be able to look at those data independently and draw conclusions themselves. But I think that, from a centralized point of view, Steve Piccoli and the others are going to provide very, very clear definition of what was like, what was not alike. So thanks for bringing that up.
Great. Thanks, Gary. Anything else?
I like the idea of a cross-company advisory panel or advisory board, which can help. We've had a couple of those instances within PSTC, different PSTC companies asking each other for advice about how to use these biomarkers. Always happy to provide that service.
Thanks, Stefan. Well, if there's no other comments, I'm going to wrap up the panel and thank the panelists and thank the speakers. I think we had a great conversation today. Laura, if we could advance the slides. This is the point at which the panelists would be hearing applause if we were actually in a real meeting. But you guys did great. Thank you. I would like to alert everybody that there are additional webinars that are coming up. Alternative solutions and safety assessment, the use of safety biomarkers beyond just safety and seeing how they can be used in disease, a session on data sharing, and then, different ways that the PSTC has influenced the formation of other consortia and other activities that folks can get involved in. And so these 4 sessions, you can see the dates for them. You can go to the website and register for those. Everyone's definitely invited. Hopefully they'll be as good as this session, because I really love this session. It was a lot of fun. And so if we can go to the final slide, Laura, and that's should be the thank you slide. And so thanks everybody for joining. Really appreciate it. If indeed you do have any questions, don't hesitate to contact CPATH, myself, or any of the CPATH staff. Look on our website. All our emails are there. So thanks everybody, and hopefully see you at the next webinar.